Okay, we're going to have a look at what an enzyme is and properties of enzymes. Now, enzymes are referred to as biological catalysts, and in the next slide I'll just describe to you what a biological catalyst is, or how it works. Because basically a catalyst is something that speeds up a chemical reaction. It's not changed by the chemical reaction itself. All enzymes are proteins. So we talked before, or we've been looking at our last section at DNA and how proteins are made from the code on the DNA. So enzymes are made from DNA code. We find them in every single living cell in our body. So when we're talking then about a catalyst, we'll move on to the next slide, I'm hoping. A catalyst, as I just mentioned there before, is something that speeds up a chemical reaction, or the rate of the speed of a chemical reaction. So it means that it makes it go faster. So if we were to think in terms of a catalyst being like a chemical, and the chemical is like giving that catalyst a special shape, like this green shape that is here, then what happens is the catalyst, it takes part in a reaction. What it does here is, you'll see that there are these two molecules here, they fit onto the catalyst, and when they have fitted onto that catalyst, what happens is that a reaction takes place, and that reaction makes these two molecules bond together, and these two molecules leave the catalyst. So the catalyst is described as being unchanged. So although it helps the chemical reaction happen, it itself is not changed. It's just whatever it is that it is reacting with, it is changed. And that means that the catalyst, see this catalyst that is here, it can be used over and over again. So it can move on and do this reaction all over again with another two of these molecules that are the same, and fit the two of them together. Now, the reason that a catalyst can speed up a chemical reaction, we're going to have a look at in the next slide. If you use some of these headings that I've got up on here to help you in making your notes, then that'll help you to give you something down the side of your Cornell note-taking. So I said to you, how does a catalyst speed up a chemical reaction? Well, all chemical reactions need energy for them to take place. So we're looking here at a chemical reaction progressing along the bottom here, and we're looking at energy that is in that chemical reaction. Now you'll see here, we're looking at this line here, so there's two things that are reacting together. And for them to start to react together, to make the products which are over here, there has to be, you've got two substances, say substance A and substance B, but there needs to be something that gives them energy for them to react. And that energy is called the activation energy. It might be that you give the reaction some heat for it to start to take place, or there's other types of ways of giving chemical reactions energy. And so you give it some energy, and when it gets the energy, then the chemical reaction will start to take place. In other words, what will happen is products that are here will start to form. So you start to make products. Now, when you look at this line that has been drawn here, if we take a line from there up to here, we call that there activation energy. That is a measure of how much energy is needed to be put into a chemical reaction for it to go from the two things that are reacting to the products. When you add a catalyst, what the catalyst does, and that's this here, I should really maybe do this in a different colour, so I'll just put it in blue over this line here. This is the catalyst working along with these reactions, and you'll see that from this point to this point here, the activation energy has been lowered. So in other words, the catalyst means that less energy is needed for the chemical reaction to happen, so the reaction can happen faster. Now, there are a number of catalysts, or just chemical catalysts, and they will lower the amount of energy in a chemical reaction. But when we're talking about an enzyme, we call them biological catalysts because they have the same properties as a chemical catalyst, but they're found inside living cells. So basically, an enzyme is a biological catalyst. It speeds up chemical reactions inside the cells. And the advantage to that is that the chemical reactions that are taking place in our cells, if there weren't enzymes there, then the reactions would happen too slowly in order for us to stay alive. So the enzymes can speed up these reactions. Now, we'll just have a wee look at some examples of, or an example of an enzyme that is found in your cells. And this enzyme here is called amylase. And amylase is found in your saliva, and it's also produced by an organ, your pancreas. Now, this enzyme that's found in your saliva, it's an enzyme that breaks down a substance, and what it does is it breaks down starch. And it breaks starch, which is a huge, big molecule. It's insoluble and it's a very, very long molecule. And if you're eating, say for example, a big starchy meal, the starch molecules are too large to be absorbed into your bloodstream. So amylase breaks that starch down to smaller molecules, and it breaks the starch down to a smaller sugar. It's called maltose. So here we've got the word equation, where starch is broken down by amylase to maltose. Now we call this here the substrate. The substrate, and I'm not very good at writing with this here, so substrate is the thing that the enzyme is working on, or the substance that the enzyme works with. So for example, starch here is the substrate of amylase. And this here, the maltose, is called the product. Now you'll see at the side here I've got this little thing here called SAM, and that is a mnemonic to help you remember this enzyme, which is starch is broken down by an enzyme called amylase to maltose. Now I want to just talk just a little bit more about what we were talking about just a moment ago when we were looking at this enzyme shape and fitting onto the thing that we're reacting with. Every single enzyme that is produced, we've said they're proteins, and they all have specific shapes. So amylase has got a specific shape to it. It's determined by the amino acids that make it up. There are all sorts of other enzymes, and some more of them you'll learn, but I'm not going to do that just now in this video. But each one of them has a different type of shape. So amylase, every single amylase molecule is all a certain shape. And more specifically, they have an area on them, like here, which is called an active site. And that active site is a specific shape that will fit on to the substrate that the enzyme reacts with. So the shape of the active site on the enzyme is said to be complementary to a specific substrate. So if we think here, we've got two molecules here that fit onto this catalyst. If we imagine this as an enzyme, we can see that this part here is the active site, and these two molecules that are here, these are the substrates, and they fit onto the active site of the enzyme. And when they fit onto that active site, the reaction takes place. And in this case here, two molecules are put together. When we're talking about amylase, we could think of amylase slightly different terms. So if we just make our amylase molecule, we'll pretend it's this short sort of shape. And what amylase is working with starch. So if I represent starch, it's just a very long molecule. I'm not going to draw lots and lots of molecules out here, but if you think of, say this is a shape, that shape will fit into this active site here, and when it fits into that active site, it in actual fact breaks down the starch molecule to make smaller molecules of maltose. So we see here the enzymes will actually break down substances. So amylase does that, but there are other enzymes, and we'll look at one of these in our next video, which will actually build up molecules, or you could think of it in terms of they sort of stick molecules together. So they synthesize or they build up new products. So that would be our product here, which is our maltose, and that would be our product here, which is these two molecules that have been stuck together. And we call this the lock and key model, and I'm hoping that this will move on to the next slide here. We call this the lock and key hypothesis, with the idea being that the enzyme is said to be specific. That is, it only works on one substrate, like a lock and key. In other words, only one key will fit into a certain shaped lock. So if we look at the diagram that I've got here, we can think of this here as our enzyme, and this here as our substrates. And you'll see the enzyme and the substrate here joining together in an enzyme substrate complex. And then what happens here is these substrate molecules have been bound together to make this molecule here. That's our product. And our enzyme is left unchanged. Here it is here to react again with another lot of these molecules here. Or in this diagram that I've got here, here's an enzyme. Here are the substrates, and they in this case fit onto the active site on the substrate, and the reaction takes place and this new molecule is made. So the lock and key hypothesis. So that is why we describe enzymes as being specific. That means that the enzyme only works on one particular substrate. In other words, we talked about amylase. Amylase only breaks down starch. It doesn't break down any other molecule that exists, any other carbohydrates or any proteins or anything like that. The only thing that the amylase will work with is with starch, and that is to do with this active site shape that is there. So that is why we get this terminology about enzymes being specific. And here, hopefully we'll get on to our last slide. This is just looking at enzyme action. I did mention before in the video, the section or one of the slides that we had before, that you can get two types of reactions with an enzyme. You can either break something down, or it's a degradation reaction where something is broken down into something smaller. And amylase is an example of that, where a large molecule is broken down to a smaller molecule. And that can be, there are other enzymes that will do that, and we'll look at another couple of examples of that. You also get what is known as a synthesis reaction, and that is where you take molecules that are small and you build them into something bigger. So that is a summary of enzyme properties. The summary being that an enzyme is a biological catalyst. It does that by lowering the activation energy of the chemical reaction that's taken place. An enzyme takes part in a reaction, but it is unchanged by the reaction itself. The thing that an enzyme works on is called the substrate, and the thing that is produced is called the product. And you also need to know that enzymes are specific. In other words, they work only on one type of substrate. So one type of enzyme will only work on one type of substrate, and that is called the lock and key mechanism. And that's a summary of enzyme properties. So if you make sure your notes have got those kind of headings, then that will help to put together an enzyme properties. We'll move on in the next video to have a look at more specific enzymes, and then to look at some other properties of enzymes and how they react in different temperatures and pHs. Thanks for listening.